I don't see a countdown. Ah.
Okay.
Now it's green.
Yes. Hello, everybody. We're live on the webinar now. Welcome today to the webinar titled 20 Years in Getting Stronger, the Ins and Outs of SUMOylation.
Hello.
My name is Scott Schachtel, and I'm a product manager at Biotechne, and I'd like to thank everybody first here for attending today. And I'd also like to thank our featured speaker, Dr. Frauke Melchior from the University of Heidelberg, and our featured moderator, Dr. Guillaume Bossis from the Montpellier Institute of Molecular Genetics in Montpellier, France. So thank you for joining us today.
Pleasure.
Yeah, and before we move to the formal presentation and introduction, I just wanted to inform the audience that this webinar is sponsored by Biotechne, and Biotechne brings together the prestigious life science research brands of R&D Systems, Novus Biologicals, Tocris Bioscience, Protein Simple, and Advanced Cell Diagnostics to provide the scientific research community with a comprehensive and world-class product portfolio, reagents, assays, instruments, and custom manufacturing and testing services. And part of our portfolio also includes the Boston Biochem product line, which is the most comprehensive and high-quality offering of ubiquitin-related products on the market. For more information on the ubiquitin portfolio, you can go over to the right-hand side of your screen. There's a Resources tab, and if you click on that tab, there will be more information related to ubiquitin resources from R&D Systems and Biotechne, but also resources related to the speakers that are presenting today. And just a couple housekeeping items before I hand it over to Dr. Guillaume. We invite you to ask questions throughout the webinar. If you have questions for the presenters, then you can use the Ask a Question box just below the presentation screen. You'll see it right below the presentation screen, and you can type your question into that, and that gets submitted to us, and we'll be keeping track of those and doing a question and answer session directly following the presentation. So at any time during the presentation, you can enter your questions in, and we'll answer those at the end after the presentation. So also, again, in the upper right-hand corner of the screen, there's the Resources tab, so feel free to browse that during the presentation. If you click on links in there, it won't close the presentation. It'll pop out a new window. So you'll still be able to listen to the presentation as well. Okay, and I think that's all for the housekeeping items that I had. So at this point, I'll hand the control over to Dr. Guillaume Bossis, and he'll take us from there. Guillaume?
Okay, so thank you, Scott. So first, I'd like to say a few words on proteostasis, which co-organized this webinar with Biotechne. So as most of you may know, proteostasis is a European cost action, which main objective is to facilitate collaborations in the field of intracellular proteolysis, in particular the ubiquitin proteasome system, ubiquitin-like autophagy, and lysosomal systems. So it gathers more than 260 groups in 30 countries, and the last proteostasis meeting will be held in Athens from 22 to 24 February. And registration are still open, and you are all welcome to participate. So now it's a real pleasure for me to introduce Frauke Melchior. So for those who don't know her, Frauke is German. She was initially trained as a chemist, but obtained a PhD in biochemistry, and then she decided to explore again another field of biology and joined Larry Jarrett's lab in California to work on nucleocytoplasmic transport. So this is where she discovered, it was in 1996, that RanGAP was covalently modified with a protein loosely related to ubiquitin that she named SUMO. So since then, she and the group have made major contribution in the understanding of the many roles of SUMOylation, and she is now professor at the University of Heidelberg and will give us an overview on what we know on SUMO 20 years after its discovery. So as Scott said, don't hesitate to ask questions on the chat. Frauke will answer at the end of the talk. So now, Frauke, it's your turn.
Thank you very much, Guillaume. Thanks, Scott. It's a great pleasure to do this here. I have to say it's my very first webinar, so it's a little strange not to be able to run around and wave with my hands, but and to see you. But I hope it will all go well. So, ins and outs of SUMOylation. What I would like to do is, so this talk is not for the SUMO experts. I hope I have the right audience here. It's really for those of you who wonder whether SUMO is something interesting to study. And I would like to give you quite some ideas about the mechanisms, the concepts, ways of how to work with SUMOylation. And I'll tell you a little bit about the history, but not too long. So I would like to start with stressing that SUMOylation is a reversible post-translational modification whose role really is to serve as molecular switch to change protein functions. And many proteins in the cell undergo constant cycles of SUMOylation and deSUMOylation, very comparable to what you know for phosphorylation. And this is because there is a set of modifying enzymes, we will go through the details, that need ATP to couple SUMO to target proteins, and there are isopeptidases that can take SUMO off. So in every eukaryotic organism, there's at least one SUMO gene expressed. In mammals, it's typically 3, SUMO 1 and the twins SUMO 2 and 3. SUMO 1 is usually attached as a single SUMO to target proteins or attached to several individual side chains, lysine residues to targets. SUMO 2 is also known to form SUMO chains. So what SUMO is doing is it is basically changing the surface of your protein and thereby allowing your protein to acquire novel interactions or loose interactions. And that then results, depending on the individual target, in changes in localization, stability, and activity. To give you just a few precise examples, SUMOylation could, for example, inhibit an enzyme by masking an interaction surface. And one example from our own work in collaboration with TCR6 lab many years ago is an E2 ubiquitin conjugating enzyme, E225K, shown here in cyan, that acquires a SUMO here in red, and you can appreciate that the modifying group is quite large compared to the modified protein. And SUMO masks a region on the N-terminal helix of E225K that this enzyme needs to interact with the ubiquitin E1. And in consequence, SUMOylated E225K is inactive because it has lost a protein interaction. Quite the opposite is known for many proteins that are SUMOylated and that recruit an effector molecule in response to SUMOylation. These effector molecules have SUMO binding domains, regions, we come back to this later, which allow them to interact with the SUMOylated target protein much better than without SUMO. And here very many examples exist in the field of transcription, where transcription factors, transcriptional co-repressors can recruit other components onto the chromatin once they are SUMOylated. Recruitment of helicases, co-repressors, chromatin remodeling factors are many examples. SUMO can also induce a conformational change. There's only one really nice example around. It's a DNA repair enzyme that undergoes a conformational change after SUMOylation, which allows it to then let go of DNA. So in this case, the SUMO binding region and the SUMO attachment region are in the same protein, and interaction in cis allows the conformational change. And then a final example that I want to give here is that SUMOylation can sometimes trigger degradation, and this is because there is a ubiquitin E3 ligase that has 4 binding sites for SUMO, and once a protein acquires a polySUMO chain, the ubiquitin E3 ligase is recruited, ubiquitinates the substrate, and leads to its degradation. So SUMOylation can lead to degradation, but not because it itself would target a protein through the proteasome, but because it can recruit ubiquitin E3 ligases. And here some very nice famous example the oncogenic PIM L-retinoic acid receptor fusion that causes a certain form of leukemia, and arsenic treatment of patients induces polysumolation and subsequent recruitment of RNA4 and degradation. So this is work by Ron Hayes' lab, for example, UKT's lab, and many, many others. So this gives you a little bit of a flavor of what SUMOylation can do. And SUMOylation affects many different proteins in all eukaryotic cells. There's been a vast flurry of proteomic studies in the last 5 years or so, which let us appreciate that under stress conditions at least, there are at least 3,000 proteins in any given mammalian cell, for example, 3,000 proteins that are affected by SUMOylation. Under non-stress conditions, the numbers are estimated to be at least 1,000 different proteins. A very nice compilation, if you want to see whether your favorite protein is affected by SUMOylation, is a publication listed here at the bottom of the slide, Hendricks and Vertigal, compiled in 2016, virtually every single proteomic study there was, so it's a wonderful resource to check. Now, if you look at the list of proteins that have been identified as SUMO targets, you may immediately appreciate that the vast majority of these proteins are typically nuclear proteins and have roles in the context of chromatin. They may contribute to genome integrity and structure. They may contribute to transcription. This is a very large group. To nuclear transport, to nuclear body structure and function, such as the PML bodies, and so on. DNA damage is another very big topic here. But there are, of course, also proteins that have roles in the cytoplasm and for which evidence is strong that they're actually SUMOylated in the cytoplasmic compartment. There are proteins such as PTB1B, the phosphotyrosine phosphatase, the TGF-beta receptor has been reported to be SUMOylated. I-kappa-B alpha seems to be SUMOylated in the cytoplasm, but also components involved in ion transport, such as a voltage-gated potassium channel, or the calcium ATPase SERCA2 that plays a very important role, for example, in heart function, which seems to require SUMOylation for full function. So irrespective of whether you work in the nuclear compartment or the cytoplasmic compartment, chance is quite high that one of your favorite proteins is regulated at some point by SUMOylation. So before I go into the details, into the mechanisms, I would like to share with you very briefly my own personal history on SUMO's discovery, because I think there may be some interesting lessons to be learned, at least for the younger ones. Those of you who are really interested, I can refer you to the annual review article in 2000 that I wrote. At that time, it was really easy, just 100 papers dealing with SUMO. Now it's a punishment to have to write a comprehensive review with 6,000 papers. Okay, so SUMOylation SUMO, the gene for SUMO, was discovered in Escherichia initially as a suppressor for MIF2 in a multi-copy suppressor screen. The original paper by Melou and Koschland in 1995 actually doesn't even mention the word SMT3, but they describe a screen and they gave the name to the gene, SMT3. Subsequently, quite a number of labs identified mouse and human homologs by similarity to the yeast gene and called them then alternatively SMT3 or SMT3A, SMT3B, and C, and so on. Then there were a number of publications that identified SUMO as a binding partner in yeast-2 hybrid studies. There were 3 papers published, but I assume that there were many, many other labs that had also discovered SUMO as a 2 hybrid interaction partner. And because none of these studies was able to really validate a strong non-covalent interaction, SUMO was in fact considered as a false positive in yeast-2 hybrid, which turned out later to be totally wrong. SUMO was simply covalently coupled in yeast and did not non-covalently interact. So the 3 key studies that discovered SUMO in 2 hybrid and named SUMO were by Shen et al. 96, who had RAD51 and RAD52 interact with SUMO, and they called it UBL1 for ubiquitin-like 1. Body et al., Nick Body and co-workers, they used the PML protein, which I mentioned earlier already, and they discovered a small protein which they called PIC1 for PML interacting component 1. And they found that they co-localize in PML bodies, but they could not detect direct interaction. And finally, at Ye's lab, Okura et al. Reported that a small protein interacted with the FAS-APO receptor, and they named this protein centrin. They found some weak interaction, some indication for direct interaction in pull-downs, but again, that evidence was not really strong. So all of these three studies we can now understand much better. PML is very nicely SUMOylated, to give just one example, but at that time it was unclear. So the literature was relatively weak at that point. So at the same time, in several labs, Mike Martunis, Mary Dasso, and ourselves, we came from a totally different angle. We studied nucleocytoplasmic transport. Mike investigated enriched nuclear pore complexes from nuclear envelopes, and I followed earlier findings that the small GTPase RAN plays an important role in nuclear transport. And because we had found that when you arrest the GTPase in a form that cannot hydrolyze its GTP, that this blocks nuclear transport, the next logical question was to ask, where is the RAN GTPase activating protein? So we decided to make antibodies, and that was something that was in my former mentor's lab, in Larry Gerace's lab. You know, one of the most important things, develop really, really good antibodies. And that's kind of the take-home message I would really like to pass on. Make fantastic tools, then you can discover something interesting. So we made antibodies, 2 different antibodies against RanGAP, one against the full-length protein, one against the C-terminal domain. We affinity purified these antibodies from rabbit serum and then investigated different tissue culture cell lines for the presence of RanGAP to subsequently study its localization. And what we immediately noticed was that there was one band that co-migrated with recombinant RanGAP, and there was a second band that migrated 20 kilodalton larger at 90 kilodalton, both in HeLa, NRK, and 3T3 cells, but also in liver extracts. Now, at that point, that was something like 95 when we started this work. The genome was, of course, not sequenced. There was very little databases around, so we suspected everything, a second gene, alternative splice variant, whatever. The experiment that resulted in first evidence that we had a modifier was basically self-fractionation. We found that this interesting novel band enriched in nuclear envelopes from rat liver, and we solubilized these nuclear envelopes and immunoprecipitated with our affinity-purified antibodies, RanGAP. And so we could IP the 70 kD band, the expected form, the 90 kD band, and some very large protein that turned out later to be RanBP2's binding partner, RanGAP's binding partner, RanBP2. We applied micropeptide sequencing, compared the triptych peptide digest of the 70 kD and the 90 kD form. We identified novel peptides that were only in the larger form and had them sequenced by micropeptide sequencing. In the end, it turned out that this was novel sequence information that was encoded by so-called extended sequence clones that we could buy by an 11 kilodalton open reading frame that showed distant homology to SUMO. And that led me to, I think, the most important experiment in my own personal career, an experiment that was very simple and still totally exciting. We just figured that if what we had was related to ubiquitin, then we should be able to recapitulate that in the test tube in a cell extract. So what we did is we took recombinant bacterially expressed RanGAP, we took HeLa lysate, we mixed the two in the presence of ATP or in the absence of ATP, took samples and ran a time course. And what we found was really stunning. The 70 kilodalton species from bacteria was converted into a higher migrating form. And that was basically when we realized that we had a ubiquitin-related modifier. And this and many other findings by our own, by us, but also by Mike Martunis in Günter Blobel's lab, led to two publications at the end of '96 from Mike Martunis on the beginning of '97 by us, publications that reported that RanGAP requires SUMOylation to be targeted to a nucleopore complex protein called RanBP2. That was the beginning, basically, of not of SUMO, that had been shown by others beforehand, but this was the beginning of SUMOylation, and that's why we now celebrate 20 years of this modification. Okay, so that was what I wanted to tell you about the history. As I said, in the 2000 review, you can find much more details to that, but what I would like to switch to now is really tell you our current understanding of the mechanism of SUMOylation. And here are a number of really nice reviews, the most recent one by Andrea Pichler from 2017 that goes much more detailed into the mechanism than what I can do in a 45-minute seminar. So I will summarize quickly and leave out, of course, many interesting facts. So there is a single E1 conjugating enzyme called RS1-UBA2 that resembles the ubiquitin-E1 and that needs ATP to form a thioester with SUMO. It transfers SUMO to a single E2 conjugating enzyme called UBC9. That's really key. And from there, SUMO is transferred to substrates such as the RanGAP protein, and an isopeptide bond is formed with lysine in the substrate. For RanGAP SUMOylation, you really only need an E1 and an E2 enzyme, and this has to do with the fact that UBC9 can bind very stably to RanGAP. This can be reconstituted in vitro. These are E. coli purified components, the E1 enzyme, E2, RanGAP, and SUMO. And you can convert very easily a full-length 70 kilodalton untagged RanGAP to a 90 kilodalton species with catalytic amounts of the enzymes. Here you may appreciate above the RanGAP band a very faint band that is the UBA2 subunit. So this is very easy, but RanGAP is really the odd one out and not the standard target. Most proteins require SUMO E3 ligases. In the SUMO field, there are quite a number of proteins in the literature that have been reported to have SUMO E3 ligase activity, but in our mind, there's only three categories of proteins that at the moment qualify as really being E3 ligases. E3 ligases are catalysts that catalyze the transfer of SUMO from the E2 to a lysine residue on the target protein. They are not themselves used up in the reaction. They can drive over and over again conversion of a substrate to the modifier in very short time. There's one larger family. This is the Pias proteins in yeast, cis1 and cis2. This is the largest family, and it's the only one that is evolutionarily conserved from yeast to mammals and into plants. There's a vertebrate-specific protein, RanBP2. I will talk a little bit more about this in a moment, the nucleopore complex protein that binds RanGAP. And the most recent addition are the ZNF proteins, ZNF451 family members that are also specific for higher eukaryotes for the animal kingdom. So these proteins recognize the E2 and they recognize the target. They facilitate an active conformation, a closed conformation of the E2 loaded with SUMO, and they are catalysts of the reaction. So the next question that I wanted to address is really how are targets recognized? So how can a system that has only a handful of enzymes, so remember in the ubiquitin field there are at least 600 different E3 ligases. Here we speak about roughly 10 E3 ligases that are currently known. How can so many E3 ligases recognize so precisely 3,000 different proteins and regulate them at different times in different places in the cell? So this is why target recognition is one of the most interesting questions at the moment. It starts with a so-called SUMOylation consensus motif. So many proteins have a tiny little motif, a bulky hydrophobic amino acid, lysine, X, and then acidic, that can be directly seen by the E2 conjugating enzyme when it's loaded either by itself or when loaded, of course, with a thioester, so in the active form. UBC9 recognizes the bulky residue, the lysine reaches into its catalytic pocket, and the acidic residue interacts with UBC9 surface. And so UBC9 can transfer SUMO to target proteins that have such a motif. Of course, typically an E3 ligase facilitates, catalyzes this step. A big advantage when you have a protein that is SUMOylated via such a consensus sequence is the fact that a simple point mutation, a lysine to arginine conversion, really abolishes SUMOylation. And this is here shown with the RanGPase activating protein. Wild-type transfection overexpression gives rise to two bands. Transfection of the single point mutation abolishes the modification. Such a motif has been found under non-stress conditions in about 50% of all SUMO targets. Work based by lots of proteomics work, for example, by Ivo Hendricks and Alfred Vertigal's lab, by Ron Hayes' lab, and many others. This motif can be expanded a little bit, so data analysis of all the mapped SUMO sites revealed that there is an enrichment of motifs with negatively charged amino acids C-terminal of the lysine. There's a enrichment of some targets, including RanGAP, that have hydrophobic residues directly N-terminal of the lysine. And most interestingly, there are motifs that are regulated by phosphorylation. This was first shown by the heat shock transcription factor, whose heat shock dependent phosphorylation triggers SUMOylation. And here the underlying reason is that the phosphorylated motif can interact better with the very basic charged UBC9 than the unphosphorylated species. Important to note, these motifs can still not properly be predicted. One aspect of that is that really the secondary structure matters. Here I show the structure, the motif taken out of the context from a structure from Chris Lima's lab, the RanGAP motif, and you can appreciate that the hydrophobic amino acid, the lysine, and the acidic amino acid reach into the same direction, interacting with the E2. If such a motif is hidden in a helix, it is not functional, and this is something that Andrea Pichler showed when she was still in my lab, when she investigated E225K SUMOylation. So a consensus site that is part of a helix can actually not be recognized. So secondary structure matters. The second mode of target recognition goes via a SUMO interaction motif, a non-covalent SUMO interaction motif, and I will describe these motifs a little later in my talk more precisely. But you can well appreciate that a target protein can either be recognized by UBC9 or by SUMO in a UBC9 SUMO thioester. And again, this interaction can be stabilized and the transfer can be facilitated by an E3 ligase that bridges between the target and the UBC9 SUMO thioester. Now, if you have targets where SUMO basically searches for the target, picks the target, then under such circumstances, if the target has a SUMO binding motif that prefers SUMO 2 over SUMO 1, then the target may be more efficiently SUMOylated with SUMO 2 compared to SUMO 1. And this is indeed the case. One example comes from our own work in 2008. Erik Mühlmeister, a former postdoc, discovered that the ubiquitin isopeptidase, USP25, is SUMOylated on multiple distinct lysine residues, none of which mutation of any of these does not abolish SUMOylation. But USP25 has a SUMO interaction motif, and if he destroys that SUMO interaction motif, for example, here in the last lane, the leucine mutation to an alanine, completely abolishes SUMOylation. And this protein, USP25, binds to SUMO non-covalently with preference for SUMO 2 or SUMO 3, and is also modified preferentially with SUMO 2 over SUMO 3. So in this case, if you mutate the lysine residue that you have identified, for example, by mass spec, the machinery will just pick the next adjacent lysine residue and put SUMO on it. So here it is much more difficult to generate a mutation that is not screwing up the total protein because you have mutated all lysine residues, but a meaningful mutant. And here our advice would be to try to identify a SUMO interaction motif and mutate that one. Single point mutation may be sufficient. And finally, there's a very nice mechanism where it's not the E2 SUMO thioester that determines which target is modified, but a mechanism in which it's really truly the E3 ligase that brings together a target and the UBC9 SUMO thioester in an orientation that allows a particular lysine residue to enter UBC9's catalytic pocket without a particular affinity of UBC9 for the target. And such a mechanism has been very nicely documented by Chris Lima's lab. They first published in 2009 a very nice structural model how this could happen, how the yeast SUMO E3 ligase cis1 would ensure specific SUMOylation of pCNA, the DNA processivity factor, on a lysine that is not a consensus lysine. It's K164. And in that model, the E3 ligase interacts with pCNA via its so-called pinnet domain, interacts with UBC9 via a ring motif, and interacts with SUMO in the UBC9 SUMO thioester with a CTD, that, with an additional region. And if you have a look in a more recent Nature paper by the Lima lab, they have really crystallized the complex in which you see the cysteine directly adjacent to the lysine that takes over SUMO. So here in this particular mechanism, it's the proximity that is brought about by a specific E3 ligase. Here UBC9 doesn't really select the target. Okay. So our interest in the lab is RanBP2, this huge protein, 358 kilodalton protein that sits at the cytoplasmic filaments of nucleopore complexes in interphase and which is found soluble but also on the mitotic spindle and on kinetochores in mitosis. This RanBP2 protein we find particularly fascinating because it forms a stable complex with SUMOylated RanGAP and UBC9, a complex that is now a multifunctional enzyme complex with at least 2 catalytic activities and in some mammalian cells even 3 catalytic activities. This complex is on the one hand a SUMO E3 ligase. You can read all about this in a paper from 2012, which can SUMOylate target proteins in the test tube, even if it is bound to SUMOylated RanGAP and to an inactive UBC9. On the other hand, the RanBP2 RanGAP complex functions to disassemble nuclear export complexes as they come out of the nucleus. Nuclear export complexes consist of nuclear export receptors, with GTPase RAN in GTP-bound form, and cargo proteins with a nuclear export signal. These complexes coming out of the nucleus encounter the RanGAP RanBP2 complex. They bind to RanBP2 because it has interaction sites for transport receptors, and RAN binding domains will kick out the cargo, and the RAN GTPase activating protein RanGAP will induce GTP hydrolysis. Understanding this multifunctional complex, its evolution and its reasoning of combining nuclear transport or the RAN GTPase cycle with SUMOylation is really one of the long-term goals in the lab. And one hypothesis that we follow in the question, why on earth do you connect RAN GTPase cycle activities with SUMOylation activities? One hypothesis that we follow is that transport receptors and the GTPase RAN help to recruit cargo proteins to the E3 ligase, and that the disassembly process provides a window of time in which SUMOylation can occur if the cargo that they bring is really a bona fide substrate. That's, of course, only one model that we can come up with. Alternatively, the CREM1 RAN NES cargo complex is suitable to position the RanBP2 E3 ligase at places where NAS cargo is, for example, in mitosis at the kinetochore, and that the E3 ligase region can then SUMOylate proteins that are adjacent to the NAS cargo, not the NAS cargo itself. But these are all hypotheses that will require identification of the relevant cargo proteins. So this is what I wanted to tell you about the conjugation side. Isopeptidases are, of course, the opposing partners. Here we have two isopeptidases, ULP1 and ULP2 in yeast. These were originally discovered by Mark Hochstrasser. And then we have in mammalian cells 6 related proteases, the so-called SEMP proteases. We have one protein, DZ1, that forms a dimer with a catalytic cleft that seems to be specific only for a very selected target. And our lab reported a few years ago USPIL1 as a SUMO E3 ligase, SUMO isopeptidase that resides in Cajal bodies and whose function we still don't understand yet. If you want to learn more about SUMO isopeptidases, I can refer you to 2 recent reviews listed here on the slide. The next topic that I would like to address is really SUMO binding motifs. This is an area that really we expect to expand significantly in the next years. For quite some time, people have believed that there is only one SUMO interaction motif, the so-called SUMO as SIM motif, which has been categorized as class 1 SUMO interaction motif in this recent review by Andrea Pichler. This is a motif, it's a very short hydrophobic stretch that interacts with SUMO, and I show you how in the next slide, interacting with one surface of SUMO that is opposite to the typical surface that ubiquitin uses to interact with all its different ubiquitin binding proteins, the isoleucine 44 patch. But a few years later, Ruth Geist-Friedlander's lab reported that DPP9, a dipeptidyl peptidase, can interact with SUMO, one specifically on the opposite side, so on the side that is corresponding to the isoleucine 44 patch of ubiquitin. And that is also the site that UBC9, the E2 conjugating enzyme, uses to interact with SUMO 1 and with SUMO 2, the UBC9 backside interaction with SUMO. And finally, there is a ZZ domain, for example, of the GREP binding protein CBP-P300. And there was a recent NMR structure that reveals how that interacts with SUMO. And again, this is a different interaction surface shown here in red, in purple. So these 3 regions on SUMO can be used for interaction, and it wouldn't be surprising if there are more regions to be discovered. And at the moment, we know 3 different types of SUMO interacting partners. The only type that is really partially understood, at least, is the class 1. So who has SUMO interaction motifs? You find them in virtually all of the SUMO enzymes. They have SUMO binding motifs. This includes the cis1 that I just showed you, the SUMO E3 ligase from yeast that SUMOylates, for example, PCNA. That has a SUMO interaction motif. The E3 ligase RanBP2 has at least 2 SUMO interaction motifs. And ZNF451 depends on a very interesting tandem SIM that is interrupted by a short proline-containing linker that is absolutely critical for its catalytic activity. SUMO isopeptidases can bind to SUMO, UBC9 can bind to SUMO. So all the enzymes have these motifs. I also told you that quite a number of target proteins have a SUMO binding module. For example, the USP25, which has a SUMO 2 specific ZIM that facilitates SUMOylation of this protein. But most interesting are, of course, all the downstream effectors, the readers that will determine the fate of a protein that is SUMOylated. And already in my introduction, I mentioned to you that there are proteins such as RNF4, which have 4 SUMO binding domains, motifs, 4 SIMs, that can bind to polysumolated PML. Another very nice example comes from Stefan Jentsch's lab, who described initially that PCNA, once SUMOylated, binds to a helicase, SRS2. And it turns out that this helicase has a SUMO interaction motif and has a motif that can bind to PCNA. Both motifs interact only with very low affinity, but when in tandem, when together, lead to very specific interaction. And again, Chris Lima has solved structural aspects of this interaction. So the downstream effectors will be the most interesting one to study in the future, and trying to understand who has SUMO binding motifs, how they can be predicted, how they interact with SUMO, will be, I think, a major challenge. But if we look to our bigger brother, the ubiquitin field, for a long time, ubiquitin binding proteins have been dormant until it was appreciated that very low affinity interactions are actually quite relevant biologically, and now we know more than 20 different ubiquitin interacting domains, and I wouldn't be surprised if this is something that will explode in the SUMO field in the years to come. The next point I would like to start to address is regulation of SUMOylation, and SUMO binding is already the first place where one can very nicely regulate by post-translational modifications such as acetylation or phosphorylation. To understand that better, what happens, I would like to point out here with this little cartoon how a SUMO interaction motif interacts with SUMO. So a SUMO interaction motif, a SIM, a classical SIM, is a stretch of hydrophobic amino acids that inserts as a beta strand in either parallel or antiparallel orientation into the hydrophobic groove formed by the beta 2 strand and the alpha 1 helix of SUMO. And highlighted here on the slide in yellow are these hydrophobic residues on SUMO, and in blue a basic residue. And so very typical functioning SIMs have hydrophobic residues flanked by negatively charged amino acids or serine residues. And if you have a serine residue, you can phosphorylate that. And so phosphorylation of a SIM motif will then facilitate interaction with SUMO because, of course, the doubly negatively charged phosphate may then interact with positively charged residues that flank the groove on SUMO. Conversely, acetylation of these positively charged residues on SUMO will abolish interaction with a protein that has a SIM. And to just list one nice example from the literature, this is a paper by Stefan Müller, who showed an acetylation switch that regulates SUMO-dependent interactions. So that's the first example of how you can regulate in the SUMO system by simply regulating SUMO non-covalent interactions. And I mentioned regulation at the level of target recognition by, for example, phosphorylating a SUMOylation consensus sequence or phosphorylating in proximity to SUMO consensus sequences. So that brings me to the next point. If we now look not at a single target, but we look a little bit broader at the whole SUMO proteome and look into the literature, what is known about regulation, then we find regulation at least 3 individual levels. So you may have individual stories where a particular target protein is precisely regulated as cells leave the G1 phase and enter S phase, for example. PCNA is, again, the best example here. So individual proteins that respond to cell cycle or respond to signals in their SUMOylation. Then there is regulation reported of whole groups of proteins, and a classic example here is work by Stefan Jentsch, who also coined the term group SUMOylation, where he described a wave of SUMOylation in response to DNA damage on the damage site, where multiple components in the DNA repair foci are simultaneously modified. And the idea here is that all these modifications contribute to recruitment and remodeling of sites at the DNA to allow repair proteins to proceed from one step to the next. And finally, in severe stress, one can observe dramatic changes in the global SUMO proteome. And severe stress is now true really for every single model organism that you look at. This could be a plant that is exposed to salt stress or heat stress. This could be a simple tissue culture cell line that you mistreat, but it could also be ischemia in the mouse brain if you induce oxygen depletion and so on. You see a massive increase in conjugates that are typically formed by SUMO 2 and 3, which in steady state, the equilibrium is on the free side. So that raises, of course, the big question, why do you need global remodeling? Is that protective? And if so, why exactly is this protective? And if you want to understand all these different levels of fine-tuning of SUMOylation, you really need to start to think about methods that allow you to work at the endogenous level of the SUMO proteome. I would like to stress here at this point again that at least our belief is that most SUMO targets are with each other in a dynamic equilibrium due to the relative rates of modification and demodification. So many proteins will undergo rapid cycles of modification, demodification. Whether the equilibrium is on the unmodified side or the equilibrium is on the modified side, that will vary depending on how you fine-tune the rates of modifying and demodifying enzymes. Now, it also depends, of course, on how many targets are in the picture. So if you, for example, induce phosphorylation of very abundant proteins that all of a sudden become efficient SUMO substrates, that will then, in such a dynamic equilibrium, influence the SUMOylation state of all other proteins if we assume that they are with each other in a dynamic equilibrium. So if you add more targets to the picture, every target will end up getting less SUMO. Likewise, if you overexpress SUMO massively, then of course you will change the balance between the different SUMO paralogs, and you will change, of course, the equilibrium between the different players. So if you now want to start to understand SUMOylation really at physiological levels, in physiological scenarios, it becomes important to look at endogenous SUMOylation. We have now a fantastic compendium of proteins that can be SUMOylated. We have a very nice idea of at what lysine residues they can be SUMOylated. But now we want to understand when are they actually SUMOylated and under which conditions are they deSUMOylated. And so for that we developed a protocol that allows us to study endogenous SUMOylation, either as a discovery tool combined with mass spectrometry, or more frequently now as a tool that allows us to follow the state of individual proteins by IP immunoblotting. The challenge, of course, in the SUMO field is, and has always been, that most proteins are not like RanGAP. So RanGAP, you see SUMOylated forms already in the total lysate of the cell. Most proteins, for example, TRIM28, you see in steady state in the total lysate only the unmodified form. Only if you can enrich the SUMOylated species by a factor of about 1,000 compared to the unmodified species can you appreciate the mono, di, and tri-SUMOylated species. So you need a protocol that allows you to enrich specifically the SUMOylated form to an extent that your antibodies against the endogenous protein will actually detect them on a Western blot. And for that, what we use is we use denaturing cell lysis. Then we use monoclonal antibodies against SUMO 1 or SUMO 2, with which we then immunoprecipitate the total SUMO proteome. And then we elute with peptides that contain the epitopes that these antibodies recognize to clean the IP up, because of course on beads you typically have so much non-specific sticking that you always end up getting non-SUMOylated proteins as contaminations. and then you probe with your antibodies against your own protein. And that this works nicely, you can see here both from HeLa cells, but also, and this is perhaps more important for you, more convincing, also when we apply such a protocol to complicated tissues such as mouse liver. So this is the unmodified form of TRIM28 in the input, and you clearly see a SUMOylated form in the IP. So we wrote a step-by-step protocol. The antibodies are available as hybridoma from a non-profit source, the Developmental States Hybridoma Bank in Iowa, for very little money. And here I would like to point out that Mike Martunis has been very generous in donating his monoclonal antibodies to SUMO 1 and SUMO 2 long time ago to the Hybridoma Bank. We followed then when we developed a second SUMO 1 antibody and also gave it to the library. That makes this protocol financially feasible, and I think it is definitely worthwhile to do that. We apply such protocols at the moment, for example, in a study where we try to understand how EGF, the growth factor, influences the SUMO proteome. We have identified a number of proteins that undergo changes in the SUMOylation status in response to EGF. We try to understand what that means. And here I just would like to show you how effective the IP works. So these are HeLa cells, serum starved, and then treated with EGF. Samples are taken at different time points. You see the input, and of some of the proteins, such as IF2BP1, you really only see the unsumolated form. You do the IP, and you appreciate that within 15 minutes, the SUMOylation is nearly gone. We can nearly not IP IF2BP1 anymore. After 60 minutes, it comes back. I think this is something that we would probably not be able to see if we would significantly overexpress SUMO, because such a fine-tuning would probably be lost. Another example is exposure to hydrogen peroxide, where we are very interested in how oxidative stress regulates SUMOylation. And again, we can use TRIM28 to demonstrate that different cell types depend very strikingly different with respect to effects on individual proteins on the SUMOylation status, whereas HeLa cells, TRIM28 SUMOylation is lost after hydrogen peroxide. In U2S cells, at the same time point, 15 minutes, SUMOylation actually gains. What that means biologically is something to find out. Here, I just want to show you that you can actually detect the SUMOylation without overexpressing either TRIM28 or SUMO. That brings me to the last aspect that I want to quickly cover, just a few more slides. Regulation of SUMOylation at global levels. One aspect that our lab is interested in is really looking at oxidative stress. And here are now numerous reports in the literature how hydrogen peroxide can affect SUMOylation at multiple levels. It can lead to phosphorylation of a SUMO E3 ligase. It can lead to inactivation of isopeptidases, which have cysteines in their catalytic center. It can influence the phosphorylation status of hundreds of target proteins. And for us, most interesting, it can induce a disulfide between the SUMO E1 and the SUMO E2. And this is actually work that my host, Guillaume Bosses, discovered when he was a postdoc in my lab. So what Guillaume found was that the SUMO E1 and E2 enzymes are actually redox switches. They form a disulfide between their two catalytic cysteines when cells are treated with hydrogen peroxide. In the Western blot, you see a dose-response curve with UBA2 antibodies, and you see that it goes from very little cross-linking disulfide bond to significant, depending, of course, on the dose. So we followed up on this story, and here I'd just like to summarize, because, again, this has been published at least last year. So Nikola Stankovic, a postdoc in my lab, wanted to know whether that pathway that Guillaume had discovered is actually important for cellular survival. And what Nikola found actually by identifying first a redox-impaired UBC9 mutant, a mutant that can be oxidized but doesn't form a stable disulfide with the E1 enzyme, he could actually show that cells need this mechanism. They need to inactivate locally and temporarily the E1 and E2 to allow maintenance of phosphorylated ATM in DNA damage repair. Failure to form the disulfide leads to rapid dephosphorylation of ATM, and the cells have a massive problem when they have the mutant instead of the wild type. Why this is, we still have to search for. One possible model would be that a phosphatase that is normally SUMOylated and active is locally deSUMOylated because E1 and E2 are inactive, isopeptidases remain active, and this is now inactive and can therefore not dephosphorylate ATM. But of course, there are many other hypotheses that we can follow. With that, I come to my to one prospect into the future. So what do we think are the key questions in the SUMO field? In our mind, what we still need is tools to identify, better tools, more tools to identify and analyze really endogenous SUMO targets in the context of physiological processes. I think there's plenty of room for really important discoveries. You know, if you find key proteins that alter the SUMOylation status, they may in turn be really important regulators for processes that we aim to understand. Of course, the question whether there are more enzymes around. Do we really only have 10 SUMO E3 ligases and a similarly small number of isopeptidases? Here, room is open for improvement. I already mentioned that I'm convinced that there will be more SUMO binding motifs to mediate downstream effects. And if we don't have more enzymes, we clearly need more regulatory mechanisms. So crosstalk with phosphorylation, acetylation, oxidation is certainly something that may contribute to specificity in the SUMO pathway. Then, of course, a question that nobody really addresses at the moment, but I think is really exciting, is does SUMO form diverse types of chains? What we all know is that SUMO 2 and 3 can form SUMO chains via SUMO's N-terminus, but mass spec data clearly prove that every single lysine in SUMO can be used to form linkages. So, of course, the big question is, are there other SUMO chains with different functions? The question what the purpose of global changes in the SUMO proteome after stress is of course an intriguing one. And the question whether there are more disease-associated mutations in SUMOylation sites. And finally, last but not least, of course, a topic that is of high relevance to the more medically inclined, whether SUMOylation and deSUMOylation can be used effectively for medical intervention. The first ideas are around, for example, in cardiovascular subjects, in heart failure, but also in cancer. Current labs in the project I've listed on the slide in case you're interested. Postdoc applications are, of course, always very welcome. And with this, I come to the acknowledgement. There have been many, many wonderful people in my lab that have left me over the last years, some of which I mentioned: Andrea Pichler, Guillaume, of course, Achim Werner, who took a recent position at the NIH. He worked on the ubiquitin subject, so that's why he wasn't even mentioned at the moment. Ruth Friedlander, who has her own group in Göttingen. I have lots of a number of current lab members whose work I highlighted throughout the talk. Nikola Stankovic did the most recent work on the oxidative stress regulation. Annette Floto, Rishikesh Das, Sebastian Richter, Tobias Ritterhof were involved in all these aspects of nuclear transport linked to SUMOylation. And of course, we wouldn't have been successful at all without wonderful collaboration partners, for example, Titia Sixma, Brenda Schulman, and many others. Thank you very much. I'm happy to answer questions.
Okay, so thank you, Frauke. So we start with one question we received now, so you can still ask questions. So it concerns oxidative stress, and it's asked why oxidative stress-induced inhibition of SUMOylation is not present for other systems like ubiquitination or nedylation.
Okay, yeah. That's a very interesting question. So, first of all, there is an example in the literature where one particular E2, UBC4, I believe, forms a disulfide with the ubiquitin E1, very similar to what we have found for the SUMO E1, E2. It turns out that it's probably the particular way how the E1 and E2 can interact with each other that determines whether they form a stable dimer. So what Nicola has discovered in the lab when he was on search for a mutant that would abolish this oxidation, what he found that a particular residue, D100, on the surface of UBC9, that is not mimicked by residues on other E2s, that this residue is really critical to allow the E1, E2 pair to form a very stable dimer. If he mutates that to an alanine, glutathione can immediately break the bond again, and so you don't detect the dimer. So that's what we believe is that this particular aspartate blocks access of glutathione to an oxidized product once formed. So the key here is not that that the SUMO enzymes are particularly prone to oxidation, but more likely that they are, once oxidized, much more stable as the disulfide. Because the disulfide, once formed, is incredibly stable even after oxidation stress is gone. You can still detect it 2 hours after oxidation. Okay. I hope that clarifies the question.
So apparently there is no other question for the moment. So I have one actually. So you spoke about the SUMO binding proteins, and that's true, we still don't know too much, but what do you expect we will find? Are there proteins that will bind SUMO alone, such as RanGAP4, or proteins that would bind both targets and SUMO, or any combination?
Yeah. So what I believe is that I consider it very likely that we will find a number of proteins that will have specific but very low affinity for SUMO. So similar to all the different binding modules that we know in the ubiquitin field, each of which has an affinity between 1 and 100 micromolar. So these affinities are far too low to allow specific activities in cells, specificity. The system becomes specific as soon as you combine 2 or 3 low affinity binding modules with each other. And so you could have either proteins like RanGAP4 that have multiple domains of the same type to bind polysumo, or you have low affinity modules that you combine with target modules. I think we will find both of them, and my feeling is that we will probably find many that combine it with target-specific interaction modules, so that we really have specific, target-specific effector proteins in signal, like we need it in every signal transduction cascade. Once you phosphorylate something, you have an effector that does not only see the phosphate, of course. It's an effector that sees the phosphorylated target better than the non-phosphorylated target. Otherwise, you can't pass on any specific signal. And that's what I would expect.
Okay, thank you. So now we have a more technical question. What about the use of N-ethylmaleimide for preparing samples for Western blot? How it works? Is it really necessary?
Okay. N-ethylmaleimide or iodoacetamide are alkylating reagents that inactivate cysteine-containing proteins, basically. Inactivate isopeptidases, but also inactivate, of course, E1, E2, and so on. If you give, it depends very much on how you make your cell extract. If you make your cell extract already with a Lemley buffer, so fully, or 8 molar urea, for example, or 6 molar guanidinium chloride, if you make it totally denaturing, then you don't need to add NEM. But if you want to lyse your cells first in a buffer, for example, in a RIPA buffer, which is not denaturing and which is not able to inactivate isopeptidases, if you don't add any brute force isopeptidase inhibitor, you will lose all your SUMO conjugates before you even start to look at them. That's the biggest problem, and that's why you need to add NEM, and you need to add NEM in really high concentration to make sure that the enzymes are really quickly inactivated.
Okay, so next question, now they are coming out. So is there a link between EGF-dependent deSUMOylation and the H2O2-dependent one? So it's asked, this is because EGF is known to induce an intracellular increase of H2O2 through aquaporins.
Oh. So, sorry, can you repeat that question? Somehow I don't see them here on my screen.
It's asked if there is a link between EGF-dependent deSUMOylation or EGF-dependent regulation of SUMOylation and H2O2.
Ah, okay, very good question. So we are not totally sure yet. Our feeling is, of course, this was one of the things that we started to try. So the deSUMOylation that we do see of some of our candidate proteins after EGF, we have started to apply NOx inhibitors, and at the moment it looks like it may not be related. But we don't really have the full answer here yet. What is clear for those who are not so familiar with this is that dependent activation of the EGF receptor leads to activation of NADPH oxidases that can locally produce hydrogen peroxide. And so, of course, this was something that we hoped for, but the candidates that we currently have may not be affected by this pathway.
Okay. So another question, how come that PML is SUMOylated upon oxidative stress if SUMOylation is sensitive to ROS?
The balance, what I introduced to you is that there are many, many different mechanisms by which hydrogen peroxide can impinge on SUMOylation. One of the mechanisms is the inactivation of E1 and E2 enzymes, but I spoke about many other aspects, for example, inactivation of phosphatases, activation of kinases, and so on. And it's always, of course, the exact balance of how you affect the different pathways, because you will not block any of these pathways fully or irreversibly. E1, E2 inactivation is reversible. So as long as the cell has still enough capacity to glutathione, for example, to break the bond open again, you will always have some E1 and E2 that is still active. Arsenic-induced oxidative stress has been suggested by the DT lab to induce disulfide bonds in PMEL, which seem to trigger polysumolation and subsequent recognition by RNF4. That does not exclude that in other areas of the cell, the E1, E2 disulfides are formed, and depending on local concentration, that can have different effects. So you can look at that yourself. If you add hydrogen peroxide, do a dose response curve with hydrogen peroxide on 5 different cell lines, you will get 5 totally different readouts. And you look at individual targets, it's really cell type dependent, which may have to do with the balance of enzymes that you inactivate, other enzymes that you activate.
Okay, and next question, do you have any experience with new SUMO-specific aptamers or nanobodies, and how will they compare as tools with the SUMO monoclonal antibodies?
Mm-hmm. We don't have any personal experience yet. I think it's definitely worthwhile to look into that. The SUMO monoclonal antibodies have a bit of a problem in the sense that their affinity for SUMO is not so high. Not if you compare them with, for example, an anti-HA antibody, which is fantastic. The monoclonal anti-SUMO antibodies are okay. They are also specific, but you need quite a bit of material really to have a full depletion of SUMO from your cell lysate. So I hope that at some point somebody develops antibodies that have a much higher affinity. But higher affinity, of course, comes also for a price. If you have higher affinity, you will have a more difficult time to elude your antigens effectively and specifically again from the beads. Because our SUMO IP protocol works nicely not because, this is a particularly nice IP. It works nicely because we can clean up the IP so nicely with the peptide elution. And if the antibodies are too high in affinity, that's a bit more tricky. But yeah, no, we don't have personal experience, but we should really start looking into that ourselves.
Okay, so another question on purification of SUMO-related proteins. Are the tubes, so for SUMO binding entities, a great tool to study endogenous SUMOylation?
The tubes, I think, have been a really nice tool, are a really nice tool for particular aspects. I think the tubes work particularly well if you have polySUMO-related proteins, because then you create higher affinity or avidity effects. But the tubes also require, if I'm not totally mistaken, natively folded protein, at least some of them. And so we have played with the tubes early on, but for our particular purpose of really having a clean, denatured, clean SUMO proteome and not all the million binding partners for SUMO-related proteins, the tubes have not worked so well. No, the antibodies have been working much better.
Okay. So maybe last question. Is it known how the SUMOylation is regulated by the acetylation, and are there any specific cellular processes when it occurs?
There are far too many ways how SUMOylation and acetylation interplay. So here I can refer you to our annual review 2013, where we picked up on this topic, acetylation, and showed 5 or 6 very different examples how the interplay occurs. To give you just 2 examples, many proteins that are SUMOylated on a particular lysine, for those it turned out that the same lysine residue can also be acetylated, so that you have really in this case, indeed, a competition between acetylation and SUMOylation, or acetylation prevents SUMOylation, deacetylation allows it. That's one example. So a simple competition for the same lysine residue. But I also told you that the SUMO-SIM non-covalent interaction can be regulated by acetylation if you acetylate SUMO. And so there was this nice paper by Stefan Müller where he showed that if you acetylate SUMO, then it cannot bind to SIMs anymore, to a particular type of SIM. Now, HDACs and HATs interact with SUMO, can interact with SUMO. So if you have a protein that is SUMOylated and you subsequently recruit an HDAC, then that will influence the acetylation status of this protein on other lysine residues, would be another example. So there is lots of different crosstalks. There has been one oxidative stress story where HIPK2 undergoes some acetylation, SUMOylation switch in response to oxidative stress, which triggers, which way around was it? The oxidative stress triggers, I think, the acetylation, and that prevents then the SUMOylation. So the literature is really now full of examples where you have interplay between these modifications.
Okay, so I think we'll stop here. So thank you again, Frauke, for this very nice overview on SUMOylation. So I just give back to Scott so that he can conclude. Scott, are you there?
Yes. Thank you, Guillaume. And thank you, Frauke, for the great talk. I hope all the attendees enjoyed it. And everybody have a great rest of your day.
Thank you very much. Thanks a lot, Guillaume. And thank you all for listening. Goodbye.
Bye now.